Hi, I'm Zhiqiang Hu, a postdoc working in the Brenner lab at UC Berkeley. Today, I will talk about privacy time bombs in omics data. We found that some types of data that were safe to share at the time they were created have privacy risks today and will have more critical issues in the future. I will first introduce the Golden State Killer case, which was a breakthrough in forensic genetics, but it also illuminates a new type of risk for research data. The criminal committed over 100 violent crimes in the 1970s and 80s. He committed no crimes in the next 30 years. Police arrested him in 2018 and he was sentenced to life recently. How could the police identify him 30 years after his last crime? I will briefly introduce how he was caught. The police collected DNA from the crime scene in the 1980s. The DNA didn't match any data in the database at that time. They also estimated his age and locations. These features are called quasi-identifiers. The police used quasi-identifiers to solve cases, but they were not enough in this case, and that's why it remained unsolved for 30 years. The breakthrough happened in 2018 when the police did a search through the GetMatch database. GetMatch contains consumer genetics data like those from 23andMe and Ancestry DNA. People upload their own genotype data to the database to find potential relatives. The police did a similar thing. The search identified about 20 distant relatives, which inferred a common ancestor who lived at least 150 years ago. They worked with a professional genealogist to reconstruct the whole family, and they matched the quasi-identifiers with current family members and identified two compelling candidates. They collected the DNA from the trash bin of one candidate and found it was a perfect match to the criminal's DNA. In this case, it was the combination of quasi-identifiers and DNA data that leads to the re-identification of the Golden State Killer. The usability of DNA data was activated by the consumer genetics database. And this is not a unique case. Many cold cases have been solved in a similar manner. A recent work suggests that this kind of re-identification could be common. About 60% of the European descent search in GetMatch database will result in a third cousin or closer match. As we saw, quasi-identifiers contributed to the re-identification of Golden State Killer. So this table from Yaniv Ehrlich shows the number of bits of information in common quasi-identifiers. It demonstrates how powerful quasi-identifiers are for re-identification purposes. For example, the US population size can be converted to 28 bits. The combination of birth date and zip code achieves 28.6 bits, which is larger than the US population. This suggests a person can be re-identified when their birth dates and zip codes are exposed. And indeed, this is true. Previous work has reported that about 90% of the US residents can be uniquely identified using their birth dates and zip codes. Then how can biological data breach personal privacy? I will introduce a computer science term, linking attack, which describes how an attacker gains access to information by linking data from two independent databases. Here is a cartoon example. A genomic database contains genotype data and names. And a genetic study contains genetic data and phenotypes, such as disease. It should be noted here it can be any phenotype collected by the study, not necessarily a genetic disease. By matching the genotype data, the attacker will know the data from the same person. In this case, the attacker knows Alice has bipolar disorder. Neither of the two data sets alone breach privacy. It is the linkage of data that allows breach of privacy. For ISMB data, I cannot match it to genotype directly, but the raw arrays contain genetic variants, which can be used as an intermediate to match against the genotype data in another database. Because the raw reads of ISMB data contain genotype information, privacy experts recommend to only share high-level summary data. Summary data of an ISMB experiment can be a gene expression or gene recount profile. For example, in the TCGA database, gene recount data are fully public, while the raw reads are under controlled access. If you want to use the raw data, you need to apply for it and commit to appropriate use. However, previous work suggests gene expression data may also have risks. Here, I showed on the right the same research study. In this case, the researcher only released the gene expression data as recommended by the privacy experts. It is not trivial to link this gene expression with the genotype directly, but still, I'm able to compute an intermediate to enable the linking. I compute the genotypes at EQTL loss line. How can this happen? For example, an EQTL can be a common SNP located in an enhancer region upstream a gene that impacts expression. The expression can be predicted from the genotype. Now, you can do the inverse also. If you see a high expression value, you can predict that the genotype is likely to be GG, then the variance at EQTL loci might also be linked to genotype data in another database. Today, gene expression data are still fully public. You can easily download gene expression values for hundreds or thousands of individuals on TCGA or GTEx database. This is because the practical risks from previous work had been considered low for two reasons. First, previous work studying cell line data suggested that only a very small number of EQTLs could be reliably predicted. So it was not possible to uniquely identify a genome from a large number of genomes. Second, there was no large database with complete genome to match against at that time. Previous methods will not work on a database like GetMatch, which requires to use its own tool for matching genotypes. As you can see here, the linking ability was estimated to be low, but today it has changed. We re-estimated the linking ability using recent GTEx data and found it is high. Both the quality and the number of powerful QTLs increase over time. For example, by using no more than 100 EQTLs on average, the gene expression data can be uniquely matched to the correct genome from a genome library size of one population. The increased risk comes from newly generated data and also new knowledge learned from this data. Another reason that the linking risk was estimated low is because no large database exists to be matched against. We have found that consumer genetics database, which use an identity by descent algorithm to match genotypes, now can serve as a practical genome library to match against. We first compared the confidently predicted genotype to the real genome using an identity by descent approach. And this achieved a total match of 3,000 centimorgans. Self-comparison of the real genome achieved a total match of 3,500 centimorgans. No segment can be identified when comparing a random genome against a real genome. This suggested that the IBD-based linking attack is super accurate in terms of both sensitivity and specificity. We also conducted a proof of principle test. We predicted genotype from a gene expression profile and uploaded the confident predictions to a GAMmatch database and compared the predicted genome with the real genome using the GAMmatch one-to-one comparison tool. It should be noted that this analysis only used a subset of genetic variants that are genotyped by the consumer genetics companies. Self-comparison of the real genome resulted in a total match of 3,300 centimorgans, while comparing the predicted genome to the real genome resulted in a total match of 70% of all the possible match. If you compare genomes from two different persons, except identical twins, the highest similarity in principle you can get is about 50%. In this case, the attacker can confidently say this is the same person's genome. We also compare the predicted genome to an artificial offspring of the real genome, and it results in a 43% match. So in summary, a person or their relative's genome can be identified in the consumer genetics database from their gene expression data. Let's walk through a hypothetical example of how gene expression data can be used to breach privacy. We start with Bob, whose doctor recruited him for a research project on depression in 2010. The researcher, Bob was promised that his personal information would be kept private. Therefore, the researcher only released the gene expression levels together with depression status, sex, and age. Then, in 2019, Bob purchased a genetic test from 23andMe, and later he uploaded his genotypes to GAMmatch database to find potential relatives. Now, imagine a male-violent attacker trying a new business after tearing off ransomware. So he is trying to blackmail people by threatening expose that they have depression. He collects public research data about depression, including this one, and he infers genotypes from the gene expression values and submits the predicted genotypes to the GAMmatch database. The search identifies Bob's genotype and the attached information. The attacker now knows Bob had depression. He also reveals Bob's email, sex, and age. Large-scale data mining companies could pursue similar strategies to their databases of demographic information. When the research data were released in 2010, they didn't cause immediate privacy issues. But 10 years later, with the accumulation of new data and new knowledge, Bob's research data could reveal private information. Useful but different example of genetic data become revealing over time is the sperm donation. Sperm donors in 80s were promised anonymity. Now, sperm donors are being contacted by biological children, whether or not they want to be. Could people who donated sperm decades ago have anticipated this problem at the time they made a donation? We should be thoughtful about what might happen to our research data also. Turning back to research data, in addition to the gene expression, many types of molecular QTLs have been also reported. Can other type of large-scale functional dataset also be used to infer genotypes? The answer is yes. For example, the DNA methylation risk is also high, as suggested by the large number of methylation QTLs here. We also analyzed splicing and DNase hypersensitive site data and found that DNase hypersensitive site data are even more powerful than the gene expression data. data previously considered safe may not be safe today. Gene expression data were long considered safe for unrestricted sharing. The minor concerns were raised when their linkage to genome data were reported, but the risks have been considered low, first because the linking ability was estimated to be low, and second because only a very small number of genomes are publicly available to match against. Our results here show that the linking ability is now high, and the risks can be activated by the consumer genetics database. So sharing gene expression levels today have privacy issues. And it is similar for DNA methylation data and DNase hypersensitive site data. Moreover, biological data are different from other types of private data. You may not care about your credit card transaction history from 5 years ago, but genomic privacy should be preserved for a lifetime and even beyond, possibly also for offspring and relatives. We have demonstrated that privacy risks of many types of biological data manifest over time due to new data and new biological discoveries. And this broadly challenges how we meet the goals of research participants to have their data shared widely for research discovery, while also to return personal privacy of their medical information. Thank you for your attention.